So in this video lesson, we are going to be looking at recombinant DNA, which for some people can be a little confusing because it's not something you can see, and it might be kind of hard for you to visualize or picture. So there's lots of really nice videos that you can always Google or look at YouTube in order to actually see the process. Remember, this goes along with my class and the note packet that you need to take. But when you look at the name recombinant DNA, it's just what it says. We're going to recombine DNA. With recombinant DNA, this is used in genetic engineering in order to make transgenic organisms. If you remember in a previous video, we talked about transgenic organisms and how these were organisms with a different creature's DNA in it, like the glowing cats. Those cats had the jellyfish DNA in it. We recombine their DNA. So this video is going to look at how this is done. So when we talk about biotechnology, which remember is also called genetic engineering, this is where we use living organisms to create a product. So for example, we use bacteria in order to make insulin. That would be an example of biotechnology. That would be an example of genetic engineering. And the DNA that's in the bacteria that has the bacterial DNA and the human insulin gene, that would be an example of recombinant DNA. So I got to show you how, to my best, how that gets made. So again, genetic engineering, the process of making changes in the DNA code of the living organism. And we're going to use these things called restriction enzymes. I always consider restriction enzymes to be scissors. This is what we're going to use in order to cut the DNA. So restriction enzymes are one of the tools that we use in genetic engineering. They're going to cut both strands of the DNA molecule at specific base sequences. Now, what does that mean? Well, we use bacteria a lot in order to create transgenic organisms, or we might do something called splicing, or we might have something called a vector, like when we make the flu vaccine. We take the genetic information from the virus and we recombine it and we allow it to grow inside of a chicken egg. That's why people who are allergic to eggs a lot of times can't get the flu virus. So it's just different ways that we can recombine the DNA. So with restriction enzymes, I have to take, for example, the bacterial DNA and I have to take the human DNA. So let's say we're going to make a bacteria that can make insulin for humans. So I have to make sure that I can glue these pieces together like pieces of a puzzle. So I have to make sure I cut them in the same place. So different restriction enzymes are going to cut my DNA up in different ways. So just like we have these scissors. So if I were to use this scissor right here, and I cut my DNA, it's going to have a different pattern than if I cut the scissors here. It just kind of like have a straight pattern. So when we're cutting, we have to make sure that I can take the human DNA and add it to the bacterial DNA. So they have to fit together like pieces of a puzzle. So I have to make sure that I use the same scissors when I cut both types of DNA. Because if I cut my bacteria with one type of scissor or restriction enzyme, and I cut my human DNA with another type of restriction enzyme, the pieces won't match together. So I'm not going to be able to recombine the DNA. I'm not going to be able to stick the two types of DNA together. So the first thing that's going to happen is when I isolate my gene, that means I know what gene makes insulin. So I'm going to pick the scissors and I'm going to go in and I'm going to cut that insulin gene out. It's just like tearing that page out of the book. And then I'm going to glue it into another notebook. I'm going to glue it into the bacteria so the bacteria will start to make the insulin. So the first thing I'm going to do is I'm going to take my DNA sample and I'm going to mix it with the restriction enzymes. And the restriction enzymes are going to cleave or cut DNA into pieces. Now this isn't random. Just like if I gave you something and I asked you to cut it out, you're not just going to randomly start cutting the page. You're going to cut, the pattern. You're going to cut around whatever it is that I've traced on the page. Well, the restriction enzymes have to know where to cut. So there's different restriction enzymes because they start cutting at different spots. So each kind of enzyme will look for a specific location on the DNA. So it might look for any time there's two A's together, or it might look for any time there's a GAA, or it might look for any time there's a CCC. It's looking for specific patterns. It has to know where to cut. And then most of the sequences are these things called palindromes. Palindromes are words, if you hold them to a mirror, they're the same front and back. So something like mom or race car or Hannah. But think of restriction enzymes as being scissors with different patterns on them. They're going to cut things differently. Now, some things will cut straight down the middle. If you'll notice here, we have this restriction enzyme. I call it a lule. It's going to look for this pattern. Anytime there's an A, a G, a C, and a T, it's going to make a cut. And notice it's going to cut it right down the middle. Okay. That's called a blunt cut. Okay. And you guys know if you have pieces of a puzzle that have straight edges, it's very hard to get those straight edges to stick together. We tend to use things like BAM-H or Hindi, or the most common restriction enzyme is called EcoRI, because these restriction enzymes make what's called a staggered cut, or what's called a sticky end. So notice in the case of EcoRI, this one right here, it looks for this pattern. It looks for a GAA-TTC. And it's going to cut between the G and the A, and then it's going to cut over, and then it's going to come down. So when it's done cutting, it has two pieces that look like this. And you can see how these are like pieces of a puzzle, so they could stick together very easily. So when I am cutting my DNA, I have to cut the bacterial DNA with the same restriction enzyme that I'm cutting the human DNA, so I can get the same pattern. I can get the same puzzle pieces put together. So nature likes to make these things called staggered cuts. A staggered cut is also what we call like a puzzle piece. Well, you can have a blunt cut, and a blunt cut cuts something right down the middle, but it's hard to stick straight ends together in a puzzle. So nature produces or prefers to use what we call a staggered cut, as you can see in this picture. And this is going to produce sticky ends. It's going to let the ends stick together. So I have the white piece of DNA that might represent the human gene, and then I have the black piece of DNA that might represent the bacterial DNA. So I need to make sure that I cut them in the same place so I can stick them together like a piece of a puzzle. So as we have here, we have something called a plasmid. A plasmid is a circular piece of DNA that we find in a bacteria. Remember, bacteria is a prokaryote. It does not have a nucleus, so it's really easy to manipulate the DNA of a bacteria. And here I have the human gene. And this, let's just say this is the gene that codes for making insulin. So we have DNA. We know that because we have two strands. We see that we have T's in here. And remember, when we look at DNA, we divide them into groups of threes called triplets, and that tells me what ingredients I need. But let's say I want to make a bacteria that can create insulin. I have to get the human gene for insulin into this plasmid. So the first thing I'm going to have to do is I'm going to have to cut the plasmid. I got to cut it open. I got to have a place to put it. And then I'm going to have to cut the DNA, the human DNA as well. So using the restriction enzyme EcoRI, or R1, which I said is one of the most common restriction enzymes we use, it always looks for the same pattern. It looks for GAA-TTC. Always. That's what it looks for. And you can see here, so if a piece of DNA did not have GAA-TTC, nothing would get cut. I would have to use a different restriction enzyme. I'd have to use different scissors. This scissors wouldn't work because it's looking for that pattern. So notice in the plasmid, it's going to cut it at the G. You can see how it cuts it. So it's going to take your plasmid here, and it's basically going to open it up. And then down here, I have the human gene, and it's going to take this gene, and it's going to cut this part out. So then I can take the human gene and add it to the plasmid. And remember, that's what we call recombinant DNA. So now I've made these sticky ends, because now remember, these ends can stick together. So notice how the A here will match the T. This A will match this T. Okay? And then up here, this A will match this T. So it's going to allow me to stick these pieces together. So here it is. I have recombined them. What's in red is the human gene, and I have now inserted that into the plasmid. So a plasmid, remember, is a circular piece of bacterial DNA. I cut it open, and then I added the human gene into that. I can then put this back into the bacteria, and the bacteria will begin to make insulin. Now, bacteria don't need insulin, so they see it as a waste product, so they're going to release it, and then I can harvest that. And so I have to add it back, and I do so using what we call bacterial transformation. So like we have here, this is my plasmid, and that's my human piece of DNA. You can see here, I cut my plasmid open, and here I cut my human piece of DNA. And remember, I have to use the same restriction enzymes. If I use one restriction enzyme to cut the bacteria and a different type of restriction enzyme to cut the human DNA, their patterns would not fit, so I would not be able to stick them together. So then I'm going to open it back up, and then as you can see here, it allows me to put the human gene into the bacteria, and then I can reinsert that recombined DNA into the bacteria. Now this bacteria is now transgenic because it does have a different creature's DNA inside of it. Now each time the bacteria copies itself, it will copy the new gene. So most bacteria will go through binary fission, which is their form of cell division, once every 20 minutes. So I really only have to do this to one bacteria. So it doesn't take a lot of time to do this to just one bacteria, and then I just let it divide, and in several days I have thousands of these bacteria, and they all have the human gene inside of them. So we've made transgenic organisms.